Good morning, everyone. So today we are discussing about a sensory receptor, which is a important topic in central nervous system. So especially in a physiology, sensory physiology, this carries a more mark. These are the basic topics you need to understand as an undergraduate students. So let us discuss about this sensory receptors in detail in another 1 hour. Slide ಹೋಗ್ತಲ್ಲ. Coming to the objective, so today we are going to discuss under this SLOs. So first we will be discussing about introduction and then the definition of the receptor and what are the receptor types, and then classification of receptors and a bit detail about the receptor potential, and later we will discuss about the properties of receptor. We know that the survival of an organism, including human, depends on having a adequate information. So these informations from where you are getting it from the external or internal environment. So this sensory input is very important for the integrity of a personality and intellectual function. If you are not receiving this sensory information from the environment, the surroundings, so that can cause it leads to a hallucination. So what is the role of this receptor? They help us to learn about this environment. So they give a constant information. So the minute to minute information will be receiving, the CNS will be received, the nervous system will be receiving through this sensory receptor. So coming to the sensory nervous system. So what are the components of sensory nervous system? It contains the first component is the sensory receptor. So this is where whatever changes happens in the environment, that is sensed by this receptor. So and then the sensation is produced. So like touch or pressure sensations, and then so those impulses, those sensations in the form of action potential is carried through by the ascending tracts, and then it reaches the first relay station, sensory relay station, that is thalamus, and then finally for the perception, for the conscious awareness, it should reach the highest center, that is somatosensory cortex. So these are responsible, all these components of sensory nervous system are responsible for the perception of sensations. So they feed the appropriate information to the appropriate mechanisms. So this constant feeding of the information is required. Why? Because to maintain the homeostatic activities of the body. So from a stimulus to perception, if you take, so all this sensory pathway begins with the stimulus, which acts on a sensory receptor. So these receptor converts all these passive forms. So these
Stimulus, receptors, transduction,
a change in the environment. So
first
that
sensory neuron,
change in the
moving
environment
into
causes the
second sensory neuron,
transduction,
third sensory
and then
neuron,
which this
inhibition,
receptor
brain,
converts the stimulus into a neural
perception,
signal. These neural signals are carried by
sensory neurons, stimulus, receptors, transduction, first sensory neuron, second sensory, third sensory, and brain ಅಲ್ಲಿ ಇನ್ಹಿಬಿಷನ್ ಬದಲು ಅಟೆನ್ಷನ್ ಹಾಕ್ಬಿಟ್ಟು ಪರ್ಸೆಪ್ಷನ್ ಮಾಡಬೇಕು.
So all sensory pathways begins with the stimulus, which acts on these sensory receptor. So these sensory receptor converts one form of energy into the other form. So that is called transduction. So which this converts into a neural signal. So these neural signals are carried by this the afferent neuron. So we have a first, second, and third order neurons. So that's where the coding and processing will be happening. So then the perception happens at the CNS. So this can be modified or inhibited with either being an attention. So if you observe this picture, the first the impression what you are going to get is, so that is first you will see this. So the hourglass, the flower vase. So if you observe carefully, you can see the face of two human faces. Human faces, they are facing each other. So that means from the stimulus to perception, so there will be changes. Maybe with the experience, so the sensory coding and the processing. So you can see the object. You can perceive the object properly. So now coming to the sensation. So what you mean by sensation? It is nothing but a subjective awareness of stimulus, whatever stimulus. So as I already have told you, the stimulus is nothing but a change in the environment. So whatever the environmental changes. Suppose if you are sitting here and then suddenly if you hear a loud noise. So that means in a quiet environment, so that loud noise acts as a stimulus. So that awareness, the person whoever is hearing that sound, so that the sound acts as a stimulus. So you can also tell the sensation as conscious awareness of changes in the external or internal environment. So then what is a perception? The perception is the conscious awareness and interpretation of those stimuli. Suppose if you are the one object is touched on your skin. So that means there is a interpretation. So whether this object is a blunt object or a pointed object, or it is noxious, or it is the safe. So you can interpret that. So what is the nature of those objects? All these things can be interpreted. That perception, when it comes to the conscious awareness, that becomes a perception. So this occurs mainly in the cerebral cortex, that is a somatosensory cortex. Some perceptions are dependent on sensation, but not all sensations are perceived. So like there are some unconscious sensations are there, so that gets filtered out in the brainstem, so that prevents the information overload. So whatever the informations, whatever the changes that is happening, so which is not a dangerous stimulus, so that will not reach the conscious level. And also some of the vegetative functions which are happening, the metabolic activity which is happening within our body, so which will not come to a conscious perception. Conscious awareness will not be there regarding that. So you can divide the sensations into two types, that is conscious and subconscious sensation. So when it comes to the conscious awareness, so then those sensations are called conscious sensation, which is not come to the conscious level, then they are called the subconscious senses. Under the conscious sensations, we have a somatic sensations and the special sensation. In subconscious, so we have again, so there is a posture, the change of posture, the minute to minute information reaches the central nervous system through the muscle about the muscle length and tension, so that appropriately you can position or adjust your postures. And certain visceral stimulus, so these will not come under conscious sensations. Coming to the receptor proper, so that is the topic of discussion today. So the neurotransmitter and hormones. So these are the chemical substances. Already you know about the neurotransmitter hormones. So these will act on a receptor which are present on a cell membrane or within the cell. So that target in the target organ, they bring about a biological changes or a response in a body. But are we talking of this receptor? So we are talking of a sensory receptors, not of a receptors, the chemicals, substances which are acting, which will bring about a chemical changes or a response in our body. So we are talking about a receptor which is present in a central nervous system, and which will bring about the changes in the body, so based on the information it is receiving. So these the sensory receptors are a part of a neuron, and they are associated with some receptors are associated with the non-neural cells, and they pick the sensory stimuli which is required for the nervous system to has to integrate and then to give a proper response. So they have to pick up these sensory stimuli. So this is the first station, so where the sensation starts the processing, and then they are called transducer. So coming to the definition of a receptor, the receptor is a structure, so which is nothing but a specialized modified nerve endings which will detect a stimulus. So whatever may be the type of stimulus, they will detect that stimulus. This is the first point of contact. So they are also called as a biological transducer. Why they are called as a biological transducer? So because the transducer is nothing but an instrument which converts one form of or any form of energy into an electrical energy. So which is the language? So in a nervous system, mainly it understand the nerve impulse. So only in the form of action potential, so the nervous system will interpret the different meanings. So that means the sensory receptors, any form of energy. So suppose if you are pointing, if a blunt object is pointing on your skin, so that is a pressure effect will be there. So that pressure is a mechanical energy. So that mechanical energy gets converted into an electrical energy at the level of sensory receptors. That's why they are called biological transducer. They are also called as an interpreters because so they translate a different languages into an electrical languages. That is the only language a nervous system can understand. So these sensory receptors are of different types. So some sensory receptors are of a free nerve endings. So whatever the neurons, the afferent neurons which will be carrying the impulses, the action potential to the higher center. So they are the dendritic endings of these neurons form the receptor. So this, for example, for the pain sensation and the temperature sensation, so these receptors are a free nerve, naked free nerve endings. Or some receptors may be the expanded nerve endings. So these dendritic endings, so that endings are thickened. So like for example in a Merkel's disc, Raffine's end bulb, so they are mainly senses the touch pressure sensation. So that means the sensory receptor could be of a naked free nerve endings or could be expanded nerve endings or it is encapsulated. So here in encapsulation what happens? So around that unmyelinated nerve ending, so there will be a covering will be there. Multilamellar capsules will be there. So like Pacinian corpuscles for a touch pressure sensation. Or some sensory receptors may be a specialized cells. So these cells, the endings are attached to a cell, so which acts as a receptor. So for example, in rods and cones, so which are the photoreceptors? So they are the specialized receptors. And then like hair cells in a cochlea, which interpret the sound. So these are called, this could be a form of specialized cells or maybe a sense organ. The receptor along with the non-neuronal cells, they form a group and they act as a sensory receptors. So for example, taste buds. So that means the sensory receptors, either it could be a naked free nerve ending or it could be expanded nerve ending or it could be encapsulated nerve endings or it could be specialized cells or the sense organ, depending on which sensation it is carrying. So there will be the type of receptor also varies. This is the structure, the receptors which are present in the skin. So usually if you take a section of the histological section of the skin, so we have a epidermis and dermis. All these receptors are located in the dermis, so in a varying position. So depending on the type of stimulus, so maybe if it is a light touch, a particular like Meissner corpuscles get stimulated. If it is a pressure, touch pressure, then Pacinian corpuscles. If it is a warm, then a Ruffini's nerve ending. So depending on a type of a sensation, a particular type of receptor gets stimulated. Coming to the classification of receptors, so you can classify the receptors in various ways, like a type of receptor which is getting stimulated, or a type of stimulus, or maybe the location of the stimulus. So depending on the nature of the stimulus, you can classify the receptors, but there is no one classification which involves all type of receptors in one particular classification. So there is a various classifications are there, so which will involve some of the receptors into this group. So based on a modality means type of a sensation it is perceiving. So you can classify the receptors into cutaneous receptors, so which will sense the cutaneous sensations like pain receptor, touch receptors, and proprioceptors mainly that will sense for the the receptor sense for the movement and position sense. So for this the receptors are located in deep in the muscles and joints. So we can classify these proprioceptors into general and special proprioceptors. Generals mainly so through the muscles and joint receptors like muscle spindle or Golgi tendon organs which are located in and around the joint and then the tendon and muscles. Special proprioceptors like the cells, the receptors which are present in the vestibular apparatus, they will maintain the balance of the body. And then the receptor for special senses, so that will be discussed in detail with the special senses. And then the receptor for visceral senses, even the visceral organs which are present in our body, so they have the receptors are present, so like baroreceptors, chemoreceptor, osmoreceptors, and the stretch receptors which are present in the particular part of the GAT. So all these are the visceral, they'll carry the visceral sensations. Depending on a source of stimulus, if you want to classify the receptor, you can classify the receptors into telereceptors, which will detect the distant stimuli. So, like the receptors are present within an organ, sensory organ, but they receive a stimulus not immediately over the receptor, it is from the distant. So, it is distant stimuli. So, like vision and hearing, those are the telereceptors. And exteroceptors, they detect a stimulus or immediate outside of the body, like over the skin, whatever the temperature, touch. So all these are the called exteroceptors. And then there is a interoceptors, so mainly which are present within the body. So that is visceral receptors. They detect a stimuli within our body. So they are the blood pressure baroreceptors or chemoreceptors, for example. And then there is a proprioceptors. The proprioceptors, which the stimulus arising from the deeper structures. So like they mainly sense the body position and the movement, mainly from the joint and the muscle. The receptors which are present in the joint and the muscle. So you can classify the receptors based on a modality, based on a source of stimulus, and you can also classify the receptors based on the type of stimulus. Based on a type of stimulus, you can classify it into, like if it is a chemical stimulus, the chemoreceptor gets stimulated. If it is a light is the form of energy, so then the photoreceptor gets stimulated, or a temperature changes, or the deformation, the mechanical stretching can cause a can stimulate the mechanoreceptors. Or if it is a very trivial noxious stimulus, then it stimulates the nociceptors. If there is osmotic change, the osmoreceptors get stimulated. If there is a for a position and a body parts, the proprioceptors get stimulated. So depending on type of stimulus, so you can classify the receptors into the various form of receptors. Depending on the distribution of these receptors, you can classify into so general senses and special senses. General senses, they are also called somesthetic senses. They are widely distributed. The special senses mainly limited to the head. So these receptors are the distribution is very less. You can classify the receptor. Also, you can classify this receptor based on the adaptation. So, how some receptors gets adapted to a continuous, a constant stimulus. Some receptors will not show this adaptation. So, based on that, you can classify the receptors into slow adapting receptors or a tonic receptors, like a pain or a muscle spindle. Pain receptors are a proprioceptors. So they will not adapt because there is a constant information should reach the central nervous system, so that you can protect your body from the noxious stimulus, or you can change your body position according to the need. So there should be a constant. There should not be these receptors should not undergo adaptation. So whereas some receptors are rapidly adapting receptors, they are called aphasic receptors. So which is also a boon to your body. So like touch, if there is a constant pressure is there, constant touch is there, after some time, if the strength of the there is no variation in the strength of the stimulus, so that is, and it is not dangerous to the body, so they get adapted. But the information, even though there is a continuous stimulus is there, so that will not reach the conscious level, that will not reach the sensory cortex to perceive it. So these are the way of classifying the receptor based on an adaptation. You can also, there is a general classification is there. So generally when you are examining a sensory system, so usually we will follow this. The special senses, so mainly, so we will concentrate on vision, hearing, and then the taste and smell, and then there are superficial or cutaneous senses. So in a sensory system we'll be examining a pain, touch, pressure, all those things. The additional sensations and deep senses like proprioception and visceral senses. Also you can classify, generally you can classify the receptors, so based on the whether they reach the conscious level or unconscious senses. So I have told you various way of classification, but not one classification which will fit all the receptors in one way to classify. So based on various, based on the type of stimulus, based on the nature of the stimulus, based on the distribution, based on the adaptations, so we have classified the receptors into a different ways. Now coming to the electrical and ionic events which are occurring in the receptors. So how it is happening? So for that, so the Pacinian corpuscle is taken. So because this is the largest receptor, so the cutaneous receptor, so large in size, and also it is present in experimental animals. Mesentery of that, they are taken from the mesentery of the experimental animals, isolated, and then so these have a myelinated nerve endings here. Because this Pacinian corpuscle is myelinated, nerve ending is there. So that is the actual receptor. So this, the concentric lamellae which are present around this unmyelinated neurons, and also it covers the first node of the Ranvier also, which is myelinated. Once the myelination is started, so there is a first node of Ranvier is there. So that is also covered in this lamella. So this helps in enhancing the receptor potential. So these are the concentric lamella, and first node of Ranvier is also included in this lamina. What is generator or receptor potential? So this is the local potential. Already you have studied this local potential in a nerve muscle physiology. So across the nerve muscle junction, neuromuscular junction, so what type of local potential is generated? That is a end plate potential. But similarly here in a receptor, so there is a generation of receptor potential. So what do you mean by this local potential? So we know that the membrane has a membrane potential. If there is a change in the membrane potential which is occurring at the local level at that point of stimulus, so here at the receptor level it's happening, so it's called a receptor potential or a generator potential. This is studied in the Pacinian corpuscles. These receptors, the Pacinian corpuscles, are the pressure receptors. So that means these are the respond for the mechanical stimulus. So when a pressure is applied, it generates a graded potential. So we already you know the difference between a graded potential and an action potential. So the graded potential is locally it is generated, it is not propagative in nature. So either it can be depolarizing or repolarizing time. So this is and it dies down after some time. Once the stimulus is ceased, so then it cannot be propagated only at a local level where the point of stimulus is there. So there will be a potential is generated here. So if you change the pressure, that's why it is called generator potential or a receptor potential. So all means same. Generator potential, graded potential, receptor potentials, or the end plate potential, all means local potential. So if you have to tell the properties of a local potential or a general potential, you have to tell all this. It is a graded one, and then it will not follow Allah Nanla. Either it is depolarizing or repolarizing type, and it is non-propagative in nature. So this, if the pressure is, if the strength of the stimulus is increased, so the magnitude will also increases. So once it reaches a threshold, then the firing level, then it produces an action potential. So till then it is a generator or a local potential. So in a Pacinian corpuscle, if you take, So if you apply the external pressure, causes the corpuscles to deform. So there will be a deformity. It undergoes, these concentric laminas are there, they undergo a deformation. So depending on the strength of the stimulus, it undergoes a deformity like this. So depending on the pressure, so that greater the pressure, so more will be the deformation. So if this deformation is happening, so and then there is a movement at the unmyelinated nerves. So there will be bending in the unmyelinated neurons. So this causes the opening up of sodium channels in the neuron membrane, which are pressure sensitive. So because of the stretch, because of the mechanoreceptors, these sodium channels gets opened up, and then so an increase in a pressure causes the sodium channels to open. So the pressure causes the sodium channels in a neuronal membrane to open up. Once the sodium channels opens up, so the sodium ion diffuses into the sodium influx will be there. So then that sodium influx occurs down the concentration gradient. So that means in ECF it is more, the sodium concentration, in ICF the concentration of sodium is less. So down the concentration gradient, because of the opening of these sodium channels, so because these are stretch sensitive channels, they get opened up, and then there is a creating a generator potential, that creates a generator potential. If the greater the pressure, the more sodium channels gets opened up, so the more the concentration gradient, a large generator potential is created. If it reaches a threshold, so the action potential develops. If it is not reaching the threshold level, it will not produce an action potential. Only a generator potential is produced. So this action potential, once the action potential is produced, so that continues to develop. That means it gets propagated, and then so it passed on to the along the neuron axons of the neuron, and then passed to the higher center. The greater the pressure, greater is the frequency of the nerve impulse along the neurons. So the maximum frequency of how much maximum frequency so it can generate, so that is limited by the refractory period. So this diagram shows the, see, if the stimulus, if it is a generator potential, if the stimulus is below the threshold, it produces a generator potential. Only once it reaches the first node of Ranvier, so once it reaches the threshold, so then they produces an action potential. Till then, so there is a local potential which is generated here, so that produces locally the membrane potential changes occurs. But as the pressure increases, goes on increasing, so that means as the intensity of the stimulus increases, so then once it reaches this threshold level, so then only the action potential gets produced. So this action potential gets propagated along this neuron till it reaches the nerve ending. It gets propagated. Coming to the source of generator potential, where exactly generator potential is generated. So I have shown you in a Pacinian corpuscle. So whether it gets generated in the lamellar level, or in the unmyelinated neuron, or in the axon, part of an axon. So where exactly this generator potential is the getting generated. Suppose if the strength of the stimulus, you are seeing a graph like A, B, C, A, B, C, D. So these are the generator potential. So that means locally this generator potential, whenever the pressure is applied here, so there is a deformation in the lamella. So like this, and then so that creates a bend in the ending of unmyelinated neuron. So this is the actual receptor. So this is the extra encapsulation is there, lamellar encapsulation. So the generation of this receptor potential will be occurring at the unmyelinated neurons. So if the more the pressure, so the more will be the amplitude of this generator potential. But once it reaches this the firing level, then only the action potential gets generated. So till then there is a local potential. In a second, this thing, so they have removed these lamellar structures, and then they have stimulated the unmyelinated neuron directly. So even when you stimulate the unmyelinated neuron, the nerve ending directly, so you can see the generator potential. So depending on the strength of the stimulus, once it reaches that threshold level, so there is an action potential is also generated. So with this experiment, it shows that this lamella is just adding up. So the maybe at threshold what you are using the stimulus, so maybe enhance the sensitivity, you can increase. But so even without the presence of these lamella, you can get a generator potentials. So this is the source, that is unmyelinated neuron, bare nerve ending is the place where the generator potential is getting generated. But also as you are increasing the stimulus, so there is a action potential is also generating. So but is the generator potential and action potential both are generating at this nerve ending? So with a further experiment, so what they did is, so they blocked this first node of Ranvier with a local anesthetic or a pressure. So if you block this first node of Ranvier and then stimulate this bare nerve ending, so then you can see only, you can record only the local potential. So you can record a generator potential. There is no problem in the production of generator potential, even though if you are increasing the strength here, but it is not reaching the threshold level, and then it is not producing an action potential. So with this experiment we came to know that, so the action potential is generated at this first node of Ranvier. So then they did one more experiment, so where there is a, where they have removed or cut allowed the degeneration of these bare nerve endings, and then stimulated the neuron, but you will not see any response. Even a generator potential or a receptor potential is not generated. So with this we can come to a conclusion with this experiment. So this bare nerve ending is very essential. So this unmyelinated neuron, the neuronal ending of a receptor, is very important for the generation of receptor potential, and the first node of Ranvier is important for the generation of an action potential. So where it reaches a threshold and then it gets propagated, the action potential. Till then whatever the stimulus you are giving, so that generates only a local potential. Here with the Pacinian corpuscles, what is happening here? So the mechanical energy is getting converted to an electrical energy. If the generator potential is strong, then only the nerve fires again. So that means the action potential is generated and continues until the generator potential reaches the first node of Ranvier, and magnitude is mainly proportional to the intensity of the stimulus. If there is a subminimal or a subthreshold stimulus you are giving, only generator potential is there. If a supramaximal or a threshold stimulus if you are giving, so then only there is a action potential is generated. Now coming to till now what we have discussed is, so we have discussed about the definition of a receptor and what are the structures of receptor, different types of receptors, and then we have discussed about the classification of receptors, and we have discussed about how this generator potential or a receptor potential is generated, and what is its ionic basis and experimental evidence. Now coming to the properties of receptors. So these receptors have certain properties. So the first is the specificity of a response, and then the second property is law of adequate stimulus, and then the receptor shows an adaptation, and the receptor has a Muller's doctrine of specific nerve energies, and then law of projection, law of intensity discrimination or Weber-Fechner law, and there is a recruitment is there, and then the last is the lateral inhibition. These are the property. Usually they'll ask for a 5 marker or maybe 2 marker. So any of these particular like adaptation they might ask, or maybe the definition of a Muller's doctrine, specific nerve energy, or Weber-Fechner law, or maybe the importance of lateral inhibitions. So they can ask for a 2 marker or either for a 5 marker, or the receptor itself they can ask as a 10 marker question. So let us discuss these properties one by one. So coming to the first property, that is specificity of a response. So each receptor responds to a particular type of a stimulus. If this is a touch, the mechanoreceptors are there, so they respond to a mechanical stimulus. They will not respond for other type of a stimulus. So that means they are modality specific. So the touch receptors will give only a touch sensation. So that means they have a threshold, a lowest threshold for a touch sensation. The pain receptor or a nociceptor is specific for a pain sensation only. So that means each receptor shows a specificity of response. And also there is a law of adequate stimulus. What do you mean by this adequate stimulus? They respond maximally to a specific stimulus. So if I'm saying a touch receptor, so that touch receptors will have a lowest threshold for the touch sensation. Suppose if I give for this lowest, that means lowest threshold, these touch receptors can be stimulated at that is the adequate stimulus, that is a threshold stimulus for that particular receptors. Suppose if that threshold is increased, so they can produce a pain sensation, so that means the other receptors can get stimulated. Suppose if you put a pressure on your eyeball, so if this is a mechanical energy, so that gives a sensation of a light sensation is there, but that is not actually a adequate stimulus for a photoreceptor. Photoreceptors, the adequate stimulus is the light, so that for the light it has a lowest threshold. But pressure is also producing, because you are putting a pressure on a eyeball, that is also producing a sensation of a light, a sensation of that is also stimulating these photoreceptors, but that is not an adequate stimulus. You have to put extra threshold, extra intensity of the stimulus is more. So the particular energy to which the receptor is most sensitive is called adequate stimulus. For example, already I have given a few examples. So here the warm water stimulates a refinese end organ, so that is the receptor thermoreceptors. Also, it can also stimulate a pain receptors when the intensity of the stimulus is more. Next, the important property is the adaptation or a desensitization. So if the stimulus of a constant strength is given continuously for a period of time, after some time what happens, there is a frequency of action potential decreases gradually. So that is called adaptation. So that means it will not reach to the conscious sensation. Suppose if you are wearing a dress for a first time, so you feel that you are wearing something over your skin. After some time, you will forget that the cloth is there on the body. So otherwise, you can see that when you are wearing a spectacles for the first time, there will be a constant information. So you will be aware of that. Okay, I am wearing something over the nose. After some time, once you get used to it, so you will not feel that perception that you are wearing a spectacles. That means if there is whatever the stimulus is there, so that is not dangerous to the body, so the body get used to it. So there will be a frequency of action potential that is generated will decrease, so that becomes habituated, so that undergoes adaptation. It will not reach the conscious level. This degree of adaptation differs with the receptors. So not all receptors will show same type of adaptation. There are two types already I have discussed in a classification. The two types of adaptation you will see, that is tonic and phasic type of adaptations. So what do you mean by tonic adaptation or a tonic receptors? So this is they show a poor or a slow or incomplete adaptation. We will not say that there is no adaptation. So there is a adaptation will be there. There is some amount of adaptation, but it is very slow. So that means when the stimulus is applied here, so there is a generation of an action potential is there. So even if the same strength of stimulus continues for a certain period of time, but there is a continuous there is a generation of action potential. So these type of receptors are called as slow tonic receptors or slow adapting receptors. So this is a very important, and it is protective in nature for certain body activity or body functions. This type of receptor, suppose if there is a pain receptors or the cold, so if it gets adapted, so that there is a chances of injuring our body will be more. So it's a kind of a protective mechanism. So some receptors will show this kind of adaptation, that is tonic. Their very incomplete adaptation or a very slow adaptation is there. So like the pain, cold or a muscle spindle, baroreceptor or a chemoreceptors, they need to check constantly there is a change in a pressure or a chemical level or change in a posture. So all these changes should reach the center at a constant phase. So this is one way of the adaptation, that is a slow adapting or a tonic type of adaptation. The other one is the phasic receptor. Here these are rapidly adapting receptors. So this, if the stimulus strength is maintained constantly, after some time the frequency of action potential that is generated goes on decreasing. So that means which is reaching the higher center, which is reaching the somatosensory cortex, is very less. So between the start of the stimulus and the ending of the stimulus, if you observe, so initially there is the number of action potential that is generated is more. So later, if it thinks that so there is no harm in with stimulating these receptors, so then there will be the number of signals that is reaching the center is decreases. So that means these are the fast adapting receptors. Only when the rate of production of this action potential, when it will change, when the strength of the stimulus is changing. So only when the strength of the stimulus is changed, so then this will change. The firing rate of the action potential will also increase. But if you are maintaining a constant strength from the application till the end for a long period of duration, so then these receptors undergoes a adaptation. So the examples for this is the touch, pressure or olfactory receptors. So this is also in one way beneficial to the body. The next, the important property is the Muller's doctrine of specific nerve energy. So we know that already I have told so many times that a nerve understands only one language, that is the impulse, nerve impulse or action potential. This action potential generated is similar in any nerve. Any nerve you take 100 million nerves, so that is generated, or you will see only this language. But how our body or our nervous system will interpret it as a different sensations, like a touch, it is not a warm or it is not a pressure, like that. So because whatever the impulse that is generated in a receptor goes to a specific part of the brain. Suppose if the a part of the brain which has to interpret the touch sensation, so that only that part will get stimulated, the impulse will reach that part of the brain. So sensory pathway for each sensation is separate from the receptor to the cortical level. So if the mechanoreceptor, the touch receptors get stimulated, so the fibers which are carrying that touch sensation will carry only that touch sensation, nothing else. Okay, from the receptor till the cortical level. So whatever the pathway is there, that is destined for one particular type of sensation. So that is called Muller's doctrine of specific nerve energy. So when a nerve pathway is stimulated, whatever the sensation is produced, so that is based on the type of receptor it is stimulated. If it is a touch receptor, so you will perceive it as a touch sensation. If it is a photoreceptor, you will perceive it as a light. If it is a hair cells which are present in a cochlea, you will perceive it as a sound. So that means it is a receptor. It is a property of a receptor. Okay, so one of the important property. So that particular type of receptor is gets stimulated till the from the stimulus from the receptor to the cortex, so that neuronal fiber or a neuronal path will carry only that type of sensation. So it can be stimulated anywhere along this pathway. It produces the same type of sensation. So that is Muller's doctrine of specific nerve energy. The next important property is the law of projection. So what do you mean by law of projection? So already I have told you from the receptor level, till it goes to the till it reaches the cortex. So there is a specific nerve pathway is there. Anywhere in this pathway, if I am stimulating here, suppose this is a pathway which carries a touch sensation, so I will not feel the sensation. If I am feeling if I am stimulating the pathway at this particular point, I will feel the sensation at the receptor level where the receptor is situated, not at the point where I am stimulating. So that means it gets projected to the point where the receptor is situated. So that is a law of projection. So anywhere in this pathway from the receptor level to the cortical level, if you stimulate, suppose if I am stimulating the cortex level where it is destined for the touch sensation of the hand. So then you will not feel the sensation because I am stimulating at the cortical level. I will not feel the touch sensation at that particular point of the cortex. I will feel the sensation where the receptor is located. So that is which is the receptor which is responsible for the hand sensation, the touch sensation over the hand. So those receptor gets stimulated. So this is called law of projection. The pathway extends. We know that the sensory pathway extends from the receptor to the cortex. Anywhere along this pathway, if I am stimulating, so the sensation is referred to the site of receptor. So this is important. For example, so this is a phantom limb. You can explain the phantom limb based on this law of projection. Suppose in a for particular reason, so there is a part of a limb is removed. Suppose you take a lower limb, a part of his limb is right lower limb is removed up to the knee joint. So maybe because of an accident. So when you stimulate the receptor which are present for the great toe. So then anywhere in this pathway if you are stimulating, you will feel the touch sensations for the great toe at the receptor location. So that means even though the limb is absent, phantom means false. The limb is not there, but you feel the touch sensation at that particular point. So this is very important. Usually they'll ask in a viva also what is law of projection, what is the basis for a phantom limb. So you need to explain. So because of the specific property of this receptor, you will feel the sensation at that location of the receptor. The next important property is the law of intensity discrimination or Weber-Fechner law. So as the name itself suggests, so it depends on your intensity of a sensation, intensity of a stimulus you are giving. So depending on that, your intensity of sensation also varies. So how are we going to differentiate whether the touch is a mild or heavy? So that is called a light touch or a pressure, right? And then so whether is a pain mild, moderate or a severe. Sometimes you will say it is acute pain, sometimes it is very severe. So depending on its subjective this thing. So how I can say that how I can discriminate all these things. So based on this law, Weber-Fechner law, according to this law, the magnitude of a sensation what we are feeling is proportional to the logarithm of the intensity of the stimulus. So what is the meaning of this? So if I am feeling some sensation, so like a touch sensation, so if I want to increase it to 2 times, the 2 times if I want to increase, I have to increase the intensity of the stimulus by 10 times because it is logarithmically related. Because of this, so if I am holding a 30 grams of a weight and 300 gram of a weight, if I am adding for a 30 gram, if I am adding 10 kg of 10 kg more weight, so definitely it gives a sensation of heavy sensation will be there. But for the same thing, if I am holding a 300 kg and if I am adding another 10 kg, it will not make much difference. Okay, so that means the magnitude of the sensation, if you have to increase by 2 times, you have to increase the intensity of the stimulus or strength of the stimulus by 10 times. So then the sensation felt, the magnitude will get doubled. So this is very important. So how I can increase that magnitude of a sensation? So by increasing the intensity of the stimulus, you can increase by varying an action potential. The same receptor will be there, but the way I am the frequency of action potential that is generated can be increased, or I can increase by including number of receptors activation. So suppose if a mild stimulus, if I the 2 or 3 receptor gets activated, if I apply a more pressure or a more stimulus for a heavy sensation, if I have to feel, so then number of action potential that is generated will be more if there is no surrounding receptors get stimulated. Or I can recruit the without varying the frequency of an action potential, I can recruit more number of receptors so that gets submitted. So this is also one of the important property of a receptor is recruitment. So depending on how many receptors get stimulated, so you can vary the intensity of a stimulus and also the magnitude of the sensation felt. The next important property is the lateral inhibition. So what do you mean by lateral inhibition? Suppose if this pointed object is touching the particular part of the skin. So there are receptors are present. So these receptors, so here there are 3 receptors are present. So these 3 receptors getting stimulated by this object. Okay, so here what is happening? The central one, the receptor which is where the exactly this point is located. So where it gets stimulated more, and then the other 2 receptors, the surrounding neighboring receptors, are getting less stimulated. Why it is happening? So because of the collaterals which are the collaterals, see which is this is the receptor, so which is maximally stimulated, that sends a collateral to the neighboring receptors. So this collateral, because of this collateral, so they stimulate the interneurons which are present in between this the afferent neurons. So because of this, so there is a lateral inhibition. They inhibit the propagation of an impulse from one neuron, the afferent neuron, to the next neuron. Okay, so because of this, so the surrounding receptors which got stimulated to a lesser degree, they get inhibited, but the neurons which stimulated maximally, so that sends an impulse to the second order neuron. So that the center perceive it as the point of stimulus is like this, even though there is a number of receptor which gets stimulated at the initial point was 3, but only the one receptor is sending an action potential to the next neuron, subsequent neuron, and that reaches the center. Okay, so what is the importance? So this is one of the very, very important property of an action potential. This occurs within the CNS, central nervous system. So this process facilitates the site of stimulation. So exact localization is possible because of this, the important property. And also one more, the significance is it gives a sharpening edge to the stimulus. So I can say that the previous figure, so it is a pointed object. So maximal stimulus where it is maximal point, the localization is there. So that is the point, the exact localization and sharp localization is possible because of this lateral inhibition. So the extent of lateral inhibition is not same in all type of modalities. It is different. It varies from a type of one type to the other type of sensation. So the maximum in the touch and the vision. So they show a maximum lateral vision. So that's why when you are seeing an object, so you can see the clearly the edge of the object, a sharpening is there or a contrast is there. Because of this, the one of the important property. And also you can exactly localize where the object is pointing, so rather than identifying it vaguely. So these are the important properties of the receptor. So to conclude, so the properties of the receptors are, so the first is specificity of a response, the second one is the law of adequate stimulus, and then the adaptation, the receptor shows adaptation, and then the receptors, there is a specific doctrine, Muller's doctrine of specific nerve energy. So each, the pathway is, you can call it as a labeled line principle also. And then there's a law of projection, and then there is a law of intensity discrimination or Weber-Fechner law, and then the recruitment of the receptors are there, and then the last one is the lateral inhibition. So these are very important properties of receptor. So they're coming to the few points over the how this sensory coding occurs. The sensory codings that to create a neuronal representations of sensory stimuli. The brain has to distinguish four elementary properties of this stimulus. What are those four elementary properties are? So one is the type of modality, it should distinguish, and then the location, the intensity, and the duration. So these are very important. The type, whether it is a touch sensation or whether it is a thermal sensations, and where exactly, which part of the body this receptor getting stimulated, and the intensity, whether it is a mild, moderate, or a severe, and for how long. So these are the important one. So the modality, again the receptors, depending on the type of receptors. So the like if it is a raffinase end organ, it is a deep pressure or warm sensation, or if it is a mucinous corpuscles, light touches. So that means, so depending on the receptor, depending on the sensation, the particular receptor gets stimulated. And then the localization, so depending again on the receptive field and a receptor distribution, so you can localize whether it is a precise localization or a vague, depending on the receptor localization is there. And also the intensity and the time course. Intensity is the frequency of action potential, so that is generated, and also for how long, for a period, for a long duration or a short duration. So depending on all these things, so these are the four important property of a stimulus which needs to be integrated at the central level. So, and then the sensory coding. So first is the receptor must convey the type of information which is sending, and the second is it must send the information about the location and the receptive field which are characteristic of a receptor, and then it must convey the intensity of the stimulus. Whether stronger the signal, more will be the frequency of action potential, or more will be recruitment of the receptor. So that this sensory coding detail we are not going, because the central processing, so it is beyond the undergraduate syllabus. So we are not discussing how exactly this sensory coding or a processing occurs. Only at the basic level, the receptor level, so how it is happening, so how the receptor potential is generated, how the action potential is generated, how it is conveyed, is there any specific pathway is there, so is there any overlap is there, all these things if you learn is good. So only at the cerebral cortical level or a somatosensory level where exactly this sensory coding or a differentiation is happening, so that is beyond the undergraduate syllabus. Now word about the receptive field. What you mean by this receptive field is, it is an area of the skin whose stimulation results in a change in a firing rate of a sensory neuron. Suppose if the one particular area if you take, so three receptors are there, so those three receptors will stimulate, and then there will be some firing rate will be there. Or if there is a very vaguely present, so in that one particular area, if this is a particular area if you are taking, so depending on the maybe the receptors are present like this, so the area of a distribution is, it will be present like this. It depending on the number of receptors present, so the rate of firing of that particular sensory neurons will get stimulated. So because of that, there is a change will occur. So the area varies inversely with the density of the receptor. So it is not same in all parts of our body. Suppose if you take a fingertip, so where if you take a millimeter, so the number of receptors which are present within that millimeter will be more. So that means the receptive field for each receptor will be very small. So it is very accurate there. So over the back or a leg if you take, so the receptors are very sparsely present. So that means the receptive field for one particular receptor will be more. So depending on that, the sensation carried by this part and this part varies. So depending on this sensory of receptor, the in the central nervous system or in a sensory cortex, the area is represented. So for a fingertips, the area represented will be more compared to the area for the back and the leg. So that's why here a vague a sensation you can feel here, you can interpret very finely. If even there is a slight change is there, you can differentiate because the representation for this particular part is more, because the more number of receptors are there, so the more information reaches the central nervous system. So today we have discussed about the sensory receptor. So I have discussed, to conclude it, to summarize it, I have given the introduction slide, introduction to the nervous system, right? So what do you mean by sensory nervous system and component of nervous system, and then what is the difference between a sensation and a perception, and we have discussed about the definition of a receptor. Which receptor are we talking? Is it a biochemistry or biochemical receptors, or the receptors which are involved in the nervous system? So that's why I have given the heading sensory receptor or a neural receptors. It is not a receptor what we talk on. So when the hormones or a neurotransmitter acts on the cell membrane. And then what are the types of receptors and classification of receptors, and we have discussed in detail about the properties of receptors, and have touched only I have given a brief overview about the sensory coding and then the receptive field. So I hope all of you have understood, so, and all of you are staying healthy and safe. So please pay attention. You have to study. You should get used to this a new model, new way of teaching. So, and there will be, you need to study continuously. Without a continuous hard work is there, so it is very difficult to cope up in the end. You might, so we don't know when the lockdown will be removed. Okay, happy learning. Thank you.